Welcome back, learners. Is going to be the third part of our kcse 2016. So we reached in question 6, and so I'm going to start from question 7. Question 6 was talking about evidences of evolution. So question 7 is going to talk about the factors that hinder self-pollination. So I'm going to start by noting these factors because they're very important, so we have to know them. So the first factor that hinders self-pollination is called protandry. The second factor that hinders self-pollination is called protogeny. And then the third factor is called self-sterility. And our fourth factor is called heterostily. So this self-sterility you'll also get in some books. It can also be called incompatibility. Okay? So to get a question you're asked, explain how the following factors affect self-pollination. Protandry, this is where the male part, that is the androecium, matures faster to shed pollen grains before the female part is mature to receive the pollen grains. You'll see, this is where the male part of the flower, is also called androecium, and you must also remember that the female part is called gynoecium. So this is where the male part of the flower, androecium, matures faster than the female part. That means the male part will mature faster to shed the pollen grain before the female part is mature to receive the pollen grains. So that hinders self-pollination. And then we have this protogeny. Protogeny is the opposite of protandry. This is where now the female part will be maturing faster to shed the pollen grains before the male part has matured to shed the pollen grains. So the female part will be receiving the pollen grains, but the male part is not ready to receive the pollen grains. Then we come to, is not ready to shed the pollen grains, Now, then we come to this other factor, which is self-sterility, that we've talked about here. Now, self-sterility, this is where now the pollen grains of a flower become sterile to the stigma of the same plant. That means they cannot germinate on the stigma of the same plant. That is what is meant by self-sterility. You'll see, this is where the pollen grains of a flower becomes sterile to the stigma of the same flower. That means a flower the anthers are producing the pollen grains, but they cannot germinate on the stigma of the same flower. So they only germinate on the stigma of another flower. That is what is meant by self-sterility. And then we have this other factor, which is called heterostily. Heterostily, this is where there is different arrangement of the style. That means you may find that the stigma is longer, but the anthers are shorter. So when the anthers produces the pollen grains, they cannot land on the stigma. That is what is called heterostily. And then we come to question 8, where we are now asked, state 3 functions of Golgi apparatus. We have 3 major functions of Golgi apparatus. 3 major functions, and these ones you must note very well. There are only 3. The first one is synthesis of lysosomes. So lysosomes are synthesized in the Golgi apparatus. And then the second one is transportation of cell secretion. Transportation of cell secretion. Transportation of cell secretion. And then we have the last one, the last one, which is packaging and transportation of glycoproteins. Packaging and transportation of glycoprotein. Remember, the word must be packaging and transportation. They have to go hand in hand. So we have covered that, the functions of Golgi apparatus, synthesis of lysosomes, transportation of cell secretions, and packaging and transportation of glycoproteins. And then we go to question 9, where we are now asked, name two structures of gaseous exchange in aquatic plant. Remember, there are terrestrial plants and aquatic plants. Terrestrial plants, these are plants that mostly grow in our normal lands. And then we now have aquatic. So when you hear the term aquatic mostly means water. So when you hear aquatic, you should always be thinking about water. So if you're asked, you name two structures of gaseous exchange in aquatic plants. One of the structures for gaseous exchange in aquatic plant, remember this is aquatic. One of them is called parenchyma tissue. And then we have the cuticle. We have the cuticle. We have the parenchyma tissue. We have the cuticle, and then we have the pneumatophores. So you must remember that for terrestrial plants, they also have their structures for gaseous exchange. One of them is stomata. So stomata is found in terrestrial, and the second one is called the cuticle. Okay, and the next one is called the lenticels. So that's why you usually have, you should say we have, when you talk about types of transpiration, you have three types of transpiration: stomatal transpiration, cuticular transpiration, and lenticular transpiration. So these are structures for gaseous exchange in terrestrial plants. And when you talk about aquatic plants, these are plants that usually live in water. Their structures for gaseous exchange is parenchyma tissue, cuticle, and pneumatophores. So I hope you have enjoyed our lessons. If you are watching this channel for the first time, kindly take your time, subscribe, share, and like. I will also do another video from question 10 until we finish this session. I hope you enjoy your lesson. Wish you all the best. Thank you.